Yeah, what's up?
This is okay for this identification.
That's not necessarily true. So we'll kind of go over that a little bit today. Well, hopefully we'll go over it. So what I'll say with single nucleotide polymorphisms, what the word literally means, and it's in the name of the word, right? Don't read, like sometimes we need to just like look at what the word is saying and not try to put a definition to it. So this is one of those examples. So single nucleotide polymorphism, polymorph just means many versions. And what it means is that we can have different versions of a gene that are dependent on just one nucleotide. So that can be good or bad. Sometimes that can be deadly. So I wouldn't define it as being deadly.
So different.
Just different genes dependent on just one nucleotide change. Yep. Sometimes it's deadly, and that's what it talks about the most. But yes. Yes and no. So that's part of it, is what I would say. Yeah. All right. Let me get this other thing out. There we
We're
go.
just old.
I got contacts.
And I don't know, because I had already talked to the director of nursing over there. Because I've been doing this last year, until I haven't taken on more than the required program, attachment period as well. And they told me that, like, yeah, when you buy, basically what they told me is. Yeah, well, I'm in my last class right now.
What's happening?
They
Can't get to this because this is in the way.
But after
I'm just trying to sign into my email.
well, I will have everything done. That's like a non-routine. We're probably delivery by this semester, so I would have nothing to do the next semester. They will take me. So maybe not them. I'm separate. Sorry, it was a mistake. We meant to clean here and seven letter. Yeah, they sent me the right letter. And it took away like all the excitement from it. Because at first I was just mad, and like I didn't respond. I didn't do any emotions, then email. No one knew that it was a mistake. Then
Finally. Okay.
Yeah, first time.
What?
Awesome. Congratulations. I'll scoot over here more so you have more room.
Oh no, I'm fine right here. I just gotta be able to reach this plug in over here. I don't bite.
Well, I don't think there's much time. So you got it? Yeah. Yeah, she emailed. I don't know, here too.
Mhm.
There was some strange man outside of the classroom, stupid in one.
What? What? That's crazy.
So, well, call me, let me know what you're looking like.
Mhm.
That happens, and, be down on somebody.
It sucks if you can't like conceal and carry on campus.
Not supposed to be.
Yeah, you're also not supposed to vape on campus. That doesn't stop people either. Just like, it's like,
I feel like getting caught with it.
that might be a bigger deal. Yeah, yeah. Yeah, you can go to
Yeah.
prison for that.
It's almost as bad as taking it in the bank or
Yeah.
liquor store.
All right, let's get mine in, everybody. I'm sure you noticed when we were making our concept maps that chapter 6 is a heavy chapter. If you haven't made your concept maps for chapter 10, you will notice very quickly that chapter 10 is a heavy chapter. You will notice probably very quickly that unit 2 is harder than unit 1. You can expect that for unit 3 as well. Unit 3 will be harder than unit 2. So if this is why we kind of eased in to unit 1, and things get a little more difficult as the course progresses. So want to be mindful of that and start studying and preparing ahead of time. We know that our next exam that's coming up, I literally don't care now, I need every notification on once. The next exam that's coming up is our lab midterm. That's on October 9th for you guys. So keep that in mind. That's not that far away, right? So we've only got a little bit of time left for that one. We've got today's lab, which we have to make up lab 6 today. So it won't be a short lab. It's a short lab for everybody else except for my Tuesday, Thursday crowd. So Monday, Wednesday, I had a short day with it, but we've got 2 labs we've got to fit in today. So it's not going to be a short lab day today. Be as short as we can make it. I'll say that, but like it's probably going to take the whole time. So this is lab 6 and 8 today. Thursday will be lab 9. Next week will be labs 10 and 11. And then the next week after that, we have a review for the lab on Monday and then the lab midterm on Wednesday. That's where we are. We're already at midterms pretty much now. So be thinking about that. We're just a couple weeks away. This is week 6. So week 8 is the middle of the semester. So we're already almost there. Put that into perspective for you guys. We're not getting started anymore. We're past that point. So we're going to go ahead and get going with our Kahoot. If you're not signed in, get signed in. And I'm not wait. Remember, if you're not signed in, you can always sign in using the number at the bottom of the screen. So we're going to get going now with chapter 6. Our first question. Jessica. Your first question would go to Jessica. I don't think Jessica's online. I think it's just Cora. Okay. Right. What is the term that refers to an organism's total sum of genetic material? First question, is Braylee here? Oh, they are. Okay. So you'll be our first person here. So the organism's total sum of their genetic material is known as their genome. So can you tell me why it's not chromosome?
Because that's more of a bigger part of this cell.
So not a bigger part, but we'll say, I'll count this because we're thinking of it being kind of different. Not quite the genome, right? The genome encompasses everything that's in your chromosomes as well as your extra chromosomal DNA. And if you're like, okay, well, who has extra chromosomal DNA? Literally all of you have extra chromosomal DNA. What I mean is DNA that's outside of your chromosomes. So your chromosomes are just part of your genome. Anybody know, give an example of some DNA that would be outside of your genome? Every single human being's genome. We've talked about it already in chapter 5, yes.
Mitochondrial DNA.
Mitochondrial DNA. It's not in your chromosomes. It's extra chromosomal. It's in the mitochondria. Your chromosomes and your genes do not code for anything in your mitochondria. Your mitochondria have their own genes. So they have their own genes. That's not in your chromosomes, right? Your X's and your Y's and all that, that's your chromosomes, okay? Bacteria have one big chromosome. So remember, I talk a lot about our genes and stuff like that because you guys don't have a background on that, generally speaking. That's very like consistent, and I feel like repetition helps a lot. So if all you're going to learn about it is in physiology, then I like to try to amp that up a little bit with repetition. But most of what we're going to be talking about is the bacteria, okay? And so we need to be thinking about how the bacterial chromosomes work too. So bacteria have chromosomes that are in one big circle, or they don't have a bunch of X's. That's not how they work, okay? They have one big circular genome, one big circular chromosome, and they can have extra chromosomal means outside of the chromosome, right? They can have extra chromosomal DNA as well, and it's usually in the form of plasmids, which was our great area. And a plasmid is a circle bit of DNA that has extra genes on it. Hate to break it to you, but eukaryotes can have that too. So we don't usually see that in multicellular eukaryotes like humans, but we see it in protozoa. Okay? So these are circles of some extra genes. They're always extra and they are not required for living, but they can help living. What I mean by that is the plasmids might carry genes to help produce toxins or have genes that help with antibiotic resistance. You can live without them, but you'll live much better if you have them. And so that's what's on the plasmid. So those are not part of the chromosome, but they are part of the genome, right? So that's what the idea is. So good job getting into that. All right, next. Because you know we're about to get real heavy here real quick, because we're going to talk about replication, transcription, and translation next. All right, physical expression of a genotype. What's the physical expression of a genotype? What's the term for that? And y'all, if you pick genotype, you're going to have a problem. All right, so the picture that depicted what was going on. So this is one thing that I feel like there's genotype and there's phenotype. Your genotype is your genes and your phenotype is what's expressed. So this picture I feel like is a pretty good representation of those two things, all right? So you always get a gene from mom and a gene from dad, whether you like them or not, or want to call them mom and dad, I don't care, okay? So you have maternal genes and paternal genes whenever we have multicellular organisms like us. Bacteria don't have maternal genes and paternal genes. They literally just have the genes they got from the cell that they've divided from. But still, they still have genotype and phenotype. So you might have a gene that codes for something, but what the cell actually does is the phenotype, okay? So the genetic information is the genotype, and what the cell's actual traits and expression are is the phenotype. So genotype is the gene code. Phenotype is what's physically expressed. Physical, genome, gene. It really is just like that, okay? And this is a good explanation of it when we have multicellular organisms that reproduce sexually, where you'll have two copies of a gene. This is what we call diploid. You have two copies of a gene, like we do. One from maternal and one from paternal. Here we have for the color gene, we have maternal and paternal. Doesn't matter which one is which, but if you got two big R's, you have a red color. If you have a big R and a little r gene, and you have a pink color. If you have two little R's, and you have a white color. Your body does this too. It's why we all have different hair color, different eye color, all sorts of stuff like that. It's not as simple as this either. Sometimes you'll express both of the genes. Other times you'll just express one or the other. And that will be determined by what we aren't going to go over much in this class, which is dominance and recessiveness of genes. And things can be co-dominant and things can be co-recessive. And you don't just have one gene version. You don't just have big R or little r. We have how many different eye colors, you know what I mean? How many different hair colors, but they're all associated with the same gene maybe. We have different versions of them and how they're going to be expressed, when they're going to be expressed. Why, when you have a mom that has red hair, you don't have red hair and stuff like that is all controlled at the genetic level. So when you guys were completing your concept maps, you probably got to operons and thought like, wow, okay, so that I'm already great. This is hard. Like a lot of people don't love an operon, okay? But let me tell you, that's the baby stuff. When we talk about gene control and gene expression, that's baby talk. What happens in your genes, so we're talking about bacteria, so we're gonna keep it in the baby talk. Your genes, it is astoundingly more complicated than an operon, astoundingly. We're not even going to touch it in this class because this is microbiology. We're talking about bacteria. And I mean, just trust me, somebody that has taken many semesters at undergrad and graduate levels of cell molecular biology and genetics, just trust me, like you're be thankful that I'm not talking to you guys about how our genes are expressed at a molecular level because it is wildly more complicated than an operon. We will go over the operons today. I'll walk you through the operons today. Obviously, chapter 6 is the much more meaty of the two chapters we're going over today. So we're going to spend more time on chapter 6 because I want to get that foundation under us because chapter 8 is meaningless if you don't get chapter 6. So we're going to spend a little more time on that today. But yes, so good. Phenotype and genotype. Let's see. How many people do we have here today? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, I think. 16 and 17 online. Okay. So I can paste this out. All right, let me see. Oh, that's not what I meant to do. If I can ask any questions about any of this. I could try. I feel like it will be a risk to ask about alleles because it doesn't talk about it. So let's talk about genes. So Tawny, can you tell me what a gene is?
Part of chromosome, basic information packet.
That's good. So we'll go with that. So it's a basic information packet that's part of your chromosome. Basically, what a gene is like a gene is an information packet that codes for a specific protein or something that regulates gene expression typically. So there's 3 different versions of genes. We have genes that make proteins, genes that control expression of genes, and genes that code for RNA. Genes that code for RNA, what I mean by that is that's all they do. That RNA is RNA, and that's what the RNA does, not mRNA. That's not what we're talking about. mRNA codes for protein. What I'm talking about is tRNA and ribosomal RNA. So you're either a gene that codes for tRNA or ribosomal RNA, which are part of making protein making machinery, right? Or you're a gene that controls expression of genes, or you're a gene that makes a protein. You do not have genes that code for fats, for carbohydrates, or for anything like that. You only have genes that make proteins. You don't have genes that say when and how to build your cell membrane. You have proteins, and the proteins work with the other proteins to know how to build a cell membrane and stuff like that. So that is basically how, like literally every cell in our body can code for anything in the body. So your liver cells don't just have genes for liver cells. Your liver cells also have the genes for your eye color, your hair color, how tall you're going to be, your metabolism, and everything like that. But so why are the liver cells only liver cells? And so that's because of gene expression and control of gene expression in those tissues. So you can see now suddenly how much more complicated we must be than bacteria. Because if every single cell in your body has every single gene, they don't have different genes. It's just which genes are expressed and controlling how they're expressed and what they're doing. So in your pancreas, you have cells that are going to be making insulin, right? Well, why do those cells make insulin and the cells in your eyes not? The genes are there, but it's just because of signals that they're getting for gene expression. So all of that is being controlled, and what dictates your tissues and what dictates your organs and all of that is just activating what genes and controlling the gene expression. And like I said, you thought an operon was bad. This is wildly more difficult. A lot more information there in every single cell, and then controlling what is being expressed when and how and why and everything like that. So that's dictated by your genes and the control of the genes. The bacteria are a little bit more simple typically than what our cells will be. Cool. Getting good, good. All right, 3 parts to a DNA nucleotide. Be sure you read through each choice. DNA specifically here. Kennedy here today? Kennedy? No, Barry's here. Barry will be our next victim then. All right, so DNA nucleotides. So this is where I'm going to get into talking about DNA structure so that we can keep in mind the molecular structure of DNA. Don't separate like DNA replication and transcription and translation from our understanding of the molecular composition of DNA because it's important the whole time. So the 3 parts of a DNA nucleotide are deoxyribose sugar, phosphate, and a nitrogenous base. So we know deoxyribose sugar and ribose are basically the same sugar. So this is my question for you, Barry. How many carbons in ribose and deoxyribose? They have the same number of carbons. So when we talk about sugars, we often talk about how many carbons each sugar has.
I would just have 6, but I'm guessing.
It's 5. Yeah. So it's 5. It's 5 for that one. All right. So it's important that we actually remember that because ribose and deoxyribose being 5 carbon sugars helps dictate the structure of our DNA. So we'll start with our DNA. In DNA, I'm not going to draw the whole structure, okay? But you have a sugar phosphate backbone. And it's literally the sides of your ladder. When we talk about DNA being like a ladder, this is it unwound, but normally we see DNA as sort of like this, right? It's a helix. But when you unwind it, it's straight, okay? So what's going on the actual backbone of the ladder is the sugar phosphate backbone. I need to hold the pen differently or I'm gonna keep pressing that button. Sugar, phosphate, sugar, phosphate, sugar, phosphate, okay? The other side of the ladder is the same, but in an opposite direction. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So you'll notice the way that I drew this was on purpose. Yes, if you keep going, you might not be able to tell the direction. If I drew the actual sugar molecules, which I'm not going to sit here and do for you, it's ridiculous, number one. Number two, I'm not going to make you recognize the structures like that, okay? But be aware of what's going on with them. We have our 5 carbon sugar, which is ribose or deoxyribose. In the case of DNA, it's deoxyribose, right? And what's actually attached to the sugar, I'll ask somebody else. Who participated? This is good. All right, Brandon, what is attached to the sugar? So a nucleotide, we have it down here. Nucleotide consists of a sugar, a phosphate, and nitrogenous base. Exactly, perfect, so beautiful. So adenine, guanine, cytosine, or thymine. Said it weirder than how I usually think of it. We all have RA, how we list the ATCG. It's ATCG, yes. So the ATCG, anybody know? So let's actually just keep asking questions. Anastasia, is Anastasia here today? Okay, Anastasia. All right, so anybody know, in DNA we use ATCG. Do you know what we do in RNA? Well, wait, let her cook, folks. This is the basics. This is the basics foundation. So you must know the difference between DNA and RNA. Exactly what I was looking for. Beautiful. So that's our biggest difference. So we're using the uracil there. Now we also know that DNA is double-stranded. In our cells and in bacterial cells, typically RNA is single-stranded. In cells, typically RNA is single-stranded. And DNA is double-stranded. In viruses, that can be different, right? Remember, viruses are not cells, right? Okay, so we have our double-stranded DNA or our single-stranded RNA. We know our sugar in DNA is deoxyribose. We know in RNA it is ribose. What's the difference between deoxyribose and ribose? They remove an oxygen, literally, deoxyed. We're just removing oxygen. That's the only difference there. And then our bases, like Anastasia said, we have uracil instead of thymine. So instead of ATCG, we have AUCG. Uracil and thymine are similar, but they are different, right? So they're very similar structure, they have similar function, but they are different. And this has to do with the functionality and the way that our RNA is going to work. So this is the idea. This sugar is always attached to a base. All right, next question, Esther. All right, so how are the bases in our ladder held together? What type of bond? I'll lead in with that. So if this base is linked like this and this base is linked like this, what holds those together? What type of bond? The hydrogen bond, yep. So hydrogen bond there holds those bases, nitrogenous bases together. It's why A is attracted to, what's A attracted to? T, okay. So T, I was going to ask a question about, but I feel like that's an easy one. Most of us know A is attached to T, and then G is attached to C. Hydrogen bonding for those guys, right? I'll put that up there in case we need to refer to it later. All right, so this is the gist of how your DNA is built. We know this, we learned this in chemistry. Refreshing this should be refresher stuff. These sugars and these phosphates, you see how I drew one side, we have sugar phosphate, and on the other side it's phosphate sugar. Well, what's really going on there is those sugars are actually reversed in how they are oriented. Chemical structure orientation there, okay? This means that this backbone is going down this way, and this backbone is facing this way, okay? And this aspect, there's a term that we use to refer to the fact that they're facing in different orientations. And we'll get to the term in a second. But it's basically dictated by, again, that orientation of the sugar. We say it's a 5-carbon sugar. So this is why I brought up and kind of reiterated that it is a 5-carbon sugar. We'll count each carbon based on what they're attached to. We number them based on what each carbon is attached to. Remember that from chemistry, right? I'm not going to make you number them or even recognize which one is numbered what. But what we do need to know is they are numbered based on what they're attached to, okay? So number 1 there. Number 2, the 5th carbon is at the end in this direction, 5 carbon here. The 5th carbon is exposed at this end. And which one is at the other end? Does anybody know? The 3rd. Okay, good. So we're on the same page there. So then on the other side, the 5 is on this end and the 3 is on this end. So that's all you need to know about it. You don't need to pick out which one is the 5th or the 3rd or anything like that, or why they're numbered that way. But just know this is why we would say they are going in opposite directions. So
Will there always be like 5 on the left and 3 on the right, or as long as they're opposite?
as long as they're opposites, yes. And when we get into the replication side, so I want to set this stuff up first, so that when we talk about replication, we have some reference, right? Okay, so that'll matter when we get into replication, kind of which direction we're going in. So I'm going to talk about a term. So the term for the opposite directions, Kristen, do you know what the term is for that?
Opposite direction.
Yes, the fact that we have the 2 sides of our double-stranded DNA going in opposite directions, there was a term for that. You can look it up. I'll give you a minute, let you cook. That's going to be your question, okay?
Are we doing 6 or 7? I mean, 6 or 8?
6. Yes. So you notice how I'm calling on you guys before we're getting to the questions, because I'm hoping that the questions will reiterate what we're talking about, right? Because we must get the genetics down and we must get chapter 10 down too, because literally everything after that is governed by our genetics and our enzymes and our things like that. So we really want to get good foundation on our genetics, make sure we understand what we're doing. Because you'll notice we still haven't gotten into replication, transcription, translation. We can't get into operons until we get into that. So that's all in just chapter 6. It's just chapter 6. So that's where we start getting that idea of like chapter 6 is a heavy chapter, right? So we want to be sure we're getting that foundation down. So whenever those strands are going there, they'll be going in opposite directions, and there's a term for that. We're going to let Kristen cook with that, all right? So the next aspect of this is going to get into is going to be replication. Before we get too much into replication, I'm going to help you out with some terms relating to this. Get a different color. We will have, okay, go away, whatever. Replication, I wish this would go away. I can't, it won't, okay. Replication. All right, Gracie here today? Okay, Gracie. So what is replication? When we talk about replication in genetics, what are we referring to? What is the definition? I say definition, I don't need, general speaking, what is replication? What are we doing?
It's just the replication of the amino acids.
Close. So close.
Continuity.
So DNA. So that's it. We're literally just copying DNA. Okay. So good enough for me. Okay. Close. We're close. Yes. I know you were opening something else. And when I do multiple things, I can't. Yes, my brain does the same thing. Transcription and translation. We're going to talk about all of these major functions in a second. Replication is just copying the DNA. So I'm going to call on other people for transcription and translation. So be ready when I call on you guys, okay? Megan, where's Megan Thompson? All right, Megan, what is transcription generally?
It's copying the DNA instruction.
Copying the DNA instructions to make RNA. Yes, DNA and making usually mRNA, but it's any any RNA, but for our process, for us to have our basic understanding, we'll say messenger RNA. Basically making a working copy of the DNA gene. Okay, and we're going to get into the process of it in a moment. And then our last one that we're going to talk about today will be translation. All right, Cora, you're online with us today, but Cora, what is translation? You can type it in the chat or say it. And we'll let you cook with that for a second. Open this in case. All right, did we get to, Kristen, did we get to those opposite direction strands yet? Okay, let me know when you do. You can Google it. Yes, you can Google it. That's fine too. So translation is going exactly right, Cora. Thank you. Translation is making that messenger RNA and turning it into protein. So we know in your DNA let's see.
How did you work out the term?
So yeah, there'll be the DNA strands are basically oriented in opposite directions. There's a term for that. It's the term for DNA strands oriented in opposite directions. All right, so we got Megan. Okay. I know Caitlin's not here today, right? Caitlin didn't end up showing up. Just want to make sure I didn't miss her. Okay. Did she email me that she would be? Okay, Hannah here. Hannah not here. And Shelby. Okay, Shelby. All right, so I had a question in my mind, now I forgot it. So we'll come back to you. All right. Do you get in there, Kristen?
Yeah, is it anti?
That's it. Parallel, anti-parallel, right? So they're parallel, right? They're anti-parallel because they're in opposite directions. Okay, beautiful, perfect, you did it. And I don't call it, well, I say don't call on you again, but you're welcome to still participate, okay? Please participate still. Okay, beautiful. And again, I don't remember what I was gonna ask. It's gonna be something about this, transcription and translation. Okay, let's just move on for now, and we'll get to that stuff in a minute. I don't want to get too far ahead on it. All right, so anti-parallel DNA strands. Okay, the nucleotides, those nucleotides are linked by, we already talked about it, but how are they linked? The A to the T or the G to the C. Those bases are linked by what kind of bond? So those are hydrogen bonds. Those are hydrogen bonds. And so we see here, one thing that we talked about, this whole structure that I had drawn for you here. Why is it not letting me click? This structure here, where I put a big bubble around a S, B, and P. That's a nucleotide. So these are just repeating nucleotides. This is a nucleotide. 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 So now we can see a little bit better that anti-parallel arrangement and how they are opposite from one another, kind of in their orientation, all right? And so that's what that is. Now imagine for a second that I take a shoelace, and I always have this talk when I'm not wearing shoes that have shoelaces. But imagine you have a flat shoelace, and you start twisting it into a helix, right? If you keep twisting it past that, the helix starts to turn in on itself. And you'll even get, like, if you just keep twisting it and keep forcing it and forcing it and forcing it and forcing it and forcing it, eventually you can get that whole thing twisted up into where you'll have these like multi-helix little twists going on it and whatever. That's how your DNA is arranged. It's not in a nice big happy helix, just nice and neat like that in your body. It's like a helix that's twisted on a helix, that's twisted on a helix, that's twisted on a helix. And the way that we are binding all that up and consolidating it is with these proteins called histones. And there's multiple kinds of histones, and how they make the bundles kind of wrap up on one another. They all have their own role in that and how they're working with that DNA strand. So the histones have that ladder kind of spun up on itself. And you can also imagine too, if that's how it normally is in your cell, spun up in a helix on a helix on a helix, just continuously all like that. And now we need to open it up to get to a gene. Well, every time that you open it up, imagine this now, you have that big old bundle of shoelace and you start straightening out them from the middle. and then pushing it towards the end. And imagine that the ends don't start untwisting, but they stay stuck twisted. Now, as you're flattening out the middle part, that's going to create tension on the parts that are still twisted, right? Imagine them being held in a vice. They can't untwist. So when you start straightening out the whole thing, you create more and more and more tension towards the ends, right? So this is a problem in DNA, quite literally. What happens is whenever we start opening up the DNA, which is involving us having to untwist it to make a copy of the DNA, then we can't get to the ends very well. Literally, your cells can't get to the ends very well. The ends of your chromosomes, which is the end of your DNA, for each line of DNA that you have in your body, those chromosomes. When we get to the ends like that, we can't make copies of the ends. Not all of them. And so we lose a little bit of the end of our chromosome every time that we make a copy of our chromosome. From birth, when you started out as one cell, and you had to multiply just to be a baby, and then you had to make more cells to grow into an adult, and then you cut your hand while you're cooking, you have to make more cells to repair that, and et cetera, and et cetera. Each time that your cell has to make a copy of its DNA, you shorten the end of the chromosome. The end of the chromosome is called a telomere. You've heard of this, I know you have. We've all heard about how as you get older, your telomeres get shorter, and it is finite. You can't lengthen your telomeres. So you can only live so long. We think part of an effect of having shorter and shorter telomeres, as it gets shorter and shorter, eventually you don't have telomere anymore, and you're getting into genetic information that you're losing. And we think this might be a factor. We're not sure, but it might be a factor in why we have such an increase in cancer. You got to understand that up until like the 1800s, people only lived to 35. Literally, like that was the average age in like 1850. Most people didn't live past 35. And now that would be wild to you. That's young to die at 35 now. We feel like probably dying at 65 would be young. And look at how many more years we're adding on, how many more telomeres are getting shortened and stuff like that. It's also a problem in cloning. If I were to take one of your cells and clone you, if those telomeres were already short, we can't lengthen the telomeres for the new baby that we made of you. It gets the same telomeres. Yeah, yes. So that's an interesting problem with cloning, right? So you want to clone a creature. If you're going to clone a creature, a multicellular creature like this that has chromosomes like that, you'll want to clone them at a young age while the telomeres are still short, right? Otherwise, you're stealing years from them. So anyways, this is a problem with cloning. It's why it's a problem with cloning. And it's why you don't see, like, yes, we can clone Dolly the sheep, but it's also why Dolly the sheep don't live very long. Okay, yes. So you have probably noticed this, that science has not perfected cloning, right? That's just a fact, not yet. This is one of the problems with that, just one, right? The other problem is that the animals that we're trying to clone need to be grown in a uterus. And how do you put that into, in animals, that's one thing, but in people, how do you clone a person for scientific experimentation without, having a woman to grow that? And what woman would consent to that and stuff like that? So I'm not saying this is stuff that isn't done on the world that we live in, but it's not something that can be done regularly and openly, right? And legally, we will say, right? So that's a whole other next level, like ethical argument with cloning, I guess you could say, but it relates, right? Anyways, so that's just relating to, as you're unwinding that helix, you lose a little bit at the ends of chromosomes. Bacteria don't have this problem. Number one, bacteria, they don't have to live very long and they can multiply very quickly, but they've got one big circle. So they just copy around the circle, so they don't lose any ends. You might be like, well, that must be convenient for them. Why aren't our cells like that? But keep in mind, the bacteria have far less genes. They don't have tissues. They aren't what we would consider conscious beings that can interact with their environment intelligently and stuff like that. That's where your genes are allowing you to do those things, and they don't. And so the way that ours is set up is for our functionality. The way theirs is set up is for theirs. When you start getting into genetics of how your gametes are made, right? Your eggs and your sperm, right? And how they're made, and how they only get one copy of each chromosome. So you will double the chromosomes. You'll get, remember how in mitosis, you copy each chromosome and then you split it out. Each new egg gets the same of everything, right? That's for every one of your 2 chromosomes. You have one from the mother and one from the father. We copied them so that each new cell has one from the mother and one from the father, right? So we'll do that to make your gametes, and then we'll split them apart. So your eggs and your sperm have one version, one from your mother. That's it, not another father one. Well, another egg would have the father one, for example. That would be it. That's how we get the variety in our eggs and our sperm, because you might have all mom ones on one egg and all dad ones on one egg. But then this egg, chromosome 1 is mom, chromosome 2 is dad, and the rest, you know what I get. You get a mixing and matching of the different chromosomes and whatever. And it goes way beyond that, because as it turns out, when that egg merges with that sperm and they come together, chromosome 1 that has, I don't know, all the genes for your hair color and eye color and whatever, and chromosome 1 from the dad, same genes, can come together and from the sperm and the egg and cross over and mix and match too. So now the chromosomes that you got did not come directly from the mom and from the dad. They're already mixes and matches of from the mom and from the dad once those gametes come together to make the embryo. So it's way more complicated, and there can be mistakes whenever they cross over. You can also accidentally, during mitosis, not separate out the chromosomes the way you're supposed to, and you end up with 3 of a chromosome instead of 2 whenever you have the gametes join, which is where we get trisomy 21, which is Down syndrome. So this is way more complicated than what we're getting into, but that stuff relates to medicine that you guys will be practicing. So that's why I don't want to just leave it there. You'll talk a little bit about this stuff in physiology as well, but I feel like the more we can build on that foundation, the better you'll do. All right, cool. Let's move on. Next question, true or false? DNA polymerase builds DNA in the 5' to 3' direction. This is where we're going to be getting into our replication, making DNA copies. All righty, so this is true. So with DNA replication, so we're going to draw it like this. You've seen this. We have separate. All right, this structure that I just drew, there's a term for this structure. Shelby, what's the name of the term for this structure? You're right. Okay, so I'll help you out a little bit. I'll come back to you with another question. This is the replication fork. This is where the DNA is being opened up to make the copy. So I'll ask you another question, all right? All right, this we're gonna open this. If this one is the 5' end, that would make this one, okay, I'll ask Shelby this one. If this one's the 5' end and this is going in this direction, what end would this end be? 3, okay, easy enough. And then this one over here would be 3 exactly, and 5. Okay, so we've got our DNA strand. Beautiful, that's good enough for me, man. We're understanding how this is oriented. Anytime you want to reconnect. Well, I said that. All right, so this is how DNA replication will work. This is where we're opening up that strand. Obviously, this continues on well beyond the screen, even if we wanted to, okay? But as we're opening up, we move in the 5' to 3' direction, and that is it. You cannot add nucleotides in any other form or any other way, okay? So this is the thing with this. You also can't add nucleotides to nothing. So we'll start with a primer. It has to be added on to something. And this is at our 5' end. This is our primer, and at first, this is RNA. We'll go back and replace it later on, okay? But at first, this is RNA. All right, then we come back with our DNA and make another color here. Again, this will be the 5' end. It'll add on to the primer and move 5, and this is 3. Again, this is the 5' end, and that's the 3. And we can do this continuously. All right, Lily, is Lily here today? Lily, The continuously made strand, what's the term for that one? Exactly, the leading strand. All right, so that's made in the 5' to 3' direction. Literally, the start is at 5 and the end towards 3. And as we add on, it'll be continuing in that direction. All right. So if this is 3 to 5, can we add the same way? Can we keep feeding into the fork? Anybody? I'm not going to ask a person on this one because we'll get a little bit lost if I just keep doing that. Okay. But can we go in that same direction, feed back into that fork? Can we go in that direction? No, right, because DNA polymerase works only in 5' to 3' direction, because this would be 3 to 5. That ain't gonna work for DNA polymerase. It can't do that, okay? So what we do instead is, with our new strand, as we opened this up, we would add an RNA primer. So just, I'll draw the little dot there so we can remember that the RNA primer was there, okay? And we'll make these as it opens up. Undo that. As it opens up, we will make these, add a primer, and then add our DNA nucleotides to it in fragments. The fragments, okay, you see how this is working? And as we open it up more, we make another fragment, okay? Again, 5' to 3'. That is exactly right. So that's the Okazaki fragments. These are called Okazaki fragments. And so then I'll ask Mariah. Is Mariah here today? No. All right, Emma. What strand makes the Okazaki fragments? Yeah, exactly, the lagging strand. You probably noticed we're knocking out most of the people with chapter 6. There's a lot to cover here, so I want to make sure we're getting it all kind of taken care of. All right, so that's our leading strand and our lagging strand. Are we tracking? This is way easier to see. There is a video. You probably noticed when you were making your concept maps, whether you watch the lecture videos or not, which you know, of course, I advocate for that. But there are extra videos on there that are videos of these things. And so I feel like it's easier to see this stuff, especially translation, is way easier to see in a video form than I can ever draw. Okay. But hey, this is replication. And remember, replication, we are just copying DNA. That's it. The only time you're going to replicate is when you're making new cells. So, but do you do that all the time? Oh, constantly, right? So even right now, you probably have Okazaki fragments in your cells. That's crazy to think about, but it is true. Your cells age out and we need to make new ones, you know? And so that's happening whether you injure yourself or not, but for sure, every time you injure yourself. And for sure, whenever you are a tiny little fertilized egg and you had to multiply up into a blastocyst and then eventually into a fetus, we were doing this all the time, pretty much constantly, right? And so that was happening then too. So here we go with these fragments. These fragments cannot stay fragments. And so there's a special enzyme that seals the fragments together. Because what would happen if we left these as fragments? Your body can't work with them in fragment form. They must be one continuous strand. And so when we link the fragments together, the bond that we form will be a covalent one, but it's called a phosphodiester bond. And so there's a special enzyme that creates that bond. Let me see who we have left. Is Maishrus here? Okay, Maishrus, do you know what the enzyme's called? Yes, okay, beautiful. Okay, so that is DNA ligase. Beautiful. All right, so DNA polymerase adds on nucleotides that match up. If there was an A on the pink strand, on the blue strand, it'll add a T. If there was a C on the pink strand, on the blue strand, it'll add a G. We're following? So that's what DNA polymerase does. DNA ligase, then, if we had to make our thing in fragments like we did with the lagging strand, DNA ligase just seals those fragments together, creating a phosphodiester bond. You notice how DNA polymerase has ACE at the end. You notice how DNA ligase has ACE at the end. We talk about a lot of enzymes in the course of this semester, and a lot of them end in ACE. ACE is a dead giveaway that it is an enzyme, and enzymes are proteins. They're specifically proteins that act as catalysts to drive chemical reactions. So you learn about catalysts in chemistry. They're things that often help overcome activation energy. We will talk about that a little bit further in depth in chapter 10, okay? So, but that's a primer for chapter 10, because now we clearly have enzymes that are important in creating things and making them happen. ACEs. So in this case, ligase, DNA polymerase, helicase. Anybody tell me what helicase does? Anybody? So we know, yes, Barry.
Unwinds the DNA.
Unwinds the DNA, that's it. So we know it's in that helix in order to open it all up. And remember, it's not just in a little helix. It's a helix on a helix on a helix. Helicase helps open that up so that we can actually get to that part, that flat part of the shoelace, so we can open up the ladder there. Okay. All right, beautiful, 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 beautiful. Let's see, let's move on. All right, which enzyme connects the Okazaki fragments formed during DNA replication? We already went over it, so we should know the answer to this one. Seals the fragments together. See how we're doing on time. We're doing it right. All right, so this one's going to be ligase. Ligase seals the fragments. Well, let's see what our next question is. During transcription, ooh, we're moving into transcription. During transcription, what are we doing? It is on the screen in pink. Transcription. So Victoria, I'm going to be asking you a question next. All right, so in transcription, RNA is produced. So it has it on here, but I'm still going to ask you the second question anyway, okay? Which is what, so transcription is when RNA is produced. What is translation?
Say it one more time, sorry.
If transcription is when we're making RNA from the DNA, then what is translation? Protein, okay. And it's on the board and we talked about it, and I actually asked that question earlier, but again, I can't reiterate it enough, right? So getting our foundation on what transcription and translation are. How many of you guys get confused onto which one is which? Yeah, and it happens all the time. So we're gonna kind of go over some stuff to help you guys remember this, hopefully. Okay, imagine that in your cell is a big cookbook with any recipe for any food that you are ever gonna eat for your entire life. Ever. All right, big cookbook, right? And it's kept in your nucleus. Just think of that as your pantry. All right, we're going to keep the cookbook in your pantry, and it's so big it can't come out of the pantry, quite literally. But you can go in there and open it up. So today you're in the mood for some spaghetti and meatballs. Okay, you want to look up the recipe for spaghetti and meatballs. All right, and you make it from scratch because you're old school like that, right? So you want spaghetti and meatballs, you're gonna make it from scratch. You go into the pantry, the nucleus, you look up that recipe, that gene, okay? And you might say, okay, I'm gonna make spaghetti and meatballs. I'm gonna write down just spaghetti and meatballs because you can't take the whole cookbook out with you. It's too big. So you're just gonna copy down that one recipe. And you take that copy with you out of the pantry, out of the nucleus, and into where you're gonna be making your actual spaghetti and meatballs. That's going to be outside of the cell. It's going to be usually wherever the ribosomes are. The ribosomes can be on the rough endoplasmic reticulum or in the cytoplasm. Bacteria don't have an endoplasmic reticulum. So bacteria, it's just in the cytoplasm. For us, it's largely the rough endoplasmic reticulum. Okay, so that's the kitchen. All right. So again, in the nucleus, the pantry, the big compendium of any recipe ever. Right, you copy down that one recipe, that one gene, a working copy of the gene, or working copy of that one recipe. That is transcription. And so think of it this way: what does a scribe do? They write down something. Yes, transcription, writing it down. So think of it that way. That's where we're writing it down. So also think of it this way: DNA is in the language of a nucleic acid. It's in DNA. DNA. Na is nucleic acid. RNA, what language is it in? Nucleic acid. We're still writing it in the same language. We're just writing down part of it. So it's handwritten as opposed to typed as it was in the recipe book. So this is another way to think of it. The recipe book is all typed up in your pantry or in your genes. DNA has the blueprint for all of your genes, all in one big old book. And it stays in the nucleus. We're going to that book, opening it up to the page to find the gene that we want and writing down that gene. That is transcription. And RNA is still nucleic acid. It's just a working copy. It's like the handwritten version. Okay, so that's what the transcription, the scribe is. And it works the exact, tell me this doesn't look the same as our replication fork up here, because it works the exact same way. All right. So on this, when in transcription, you notice how we are, let me get my little thing. I'm going to write it on this picture, and you know the picture's not going to stay around, so I'll have to erase it afterwards. So pay attention to how I'm writing it, okay? All right, so this bottom strand that we are copying might look like that pink. Let me write it in the pink because it's got pink and blue here, so let's use the same colors. This is your DNA, and your DNA might have a T, A, G. Actually, let me make that a C. There's a reason I'm doing this. Hold on. Oh my gosh. A, C, and then this one might be a G, G, C. Who knows? C, and then I don't know. T, T, T. Who cares? I'm making this really pretty. Now it's getting messy because I'm thinking too fast and not acting. Anyway, so there's our DNA, and then we're looking to blue for our RNA. All right, so what's this one going to be? A. What's this next one going to be?
You.
Ah, there we go. Because we're making RNA here. This is RNA. So it's you. Yeah, everybody know why? Yes? So what's this is C, 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 G, and then A, A, A. Okay. We seeing that? So I just transcribed. So that's what's happening inside of here. But we're copying this strand here because we're making the complementary nucleotides on it. So one of these strands on that DNA serves as our template. Sorry, there's a button on the side of this pen that I keep clicking on accident. One of these strands of the DNA serves as our template. If we were to look at the other strand of DNA, instead of when we opened it up, right? So on the other side, this side up here that's open that we're not copying, what does that strand look like? Well, it looks like A, T, G, C, C, G, A, right? So that's the other DNA strand. This DNA strand we call the coding strand. It has the same code that your RNA will have, but instead of U's, it has T's on the DNA. Okay? So we have on your DNA a coding strand and a template strand. The template is the template, it's like the template for making the RNA. And then the RNA ends up having this code here. So we could say that we end up with this blue RNA as a result of transcription. When we replicated, we did the exact same thing. We just didn't use RNA nucleotides, we used DNA nucleotides, right? So when we talked about DNA replication, in the DNA above, so this is a different process, right? So in the DNA above, what we end up getting from DNA replication, you have an old strand, and you had an old strand, and you had a new strand. This is both just DNA. We're back on replication, okay? This process of DNA replication, what you'll get one old and one new on your new double helix, right? Both of them have that. That process is why we call DNA a semi-conservative process. Okay. So it's the same concept when we make our RNA, but now we're not making a copy of our DNA. We're just opening up the DNA and instead just making one single stranded RNA. But it's the same exact kind of process. So that's why I don't go into too much detail, why they don't actually cover it that much, because it's the same idea, but we're using a different enzyme. Okay. Instead of using DNA polymerase, what are we using in transcription? RNA polymerase. Well, other than that, it's the same process. And it's just going to make a single-stranded RNA. All right, so our next question goes to Mikey. So Mikey, this is going to be about transcription for you, okay? What type of RNA would we be making here? So there's 3 types of RNA. If we're making a working copy of a gene that we're going to turn into a protein, what type of RNA would that be? So I'll even help you out a little further. There's either mRNA, tRNA, or rRNA. mRNA, good. So that'll be mRNA, which is our messenger RNA. If we're making a working copy of the gene, and that working copy we're going to make a protein from, then that would be an mRNA. If we're making a copy of the gene to make a tRNA, then that would just be our result. It works the same way though. If we're making tRNA, it works the same way, but if that's your end goal is to make tRNA, then you'd be done after this. You don't go through translation. Our point is we're going through transcription to get to translation. So we're going to translate that RNA strand into a protein next. Remember, transcription, we went into the pantry, we made a copy of our recipe. Translation is coming next. We're going to make a protein out of it. All right. Everybody kind of tracking so far? Okay. So I know this should be a review, but I also am not crazy. I know that it is a lot. All right. So I'm going to erase our beautiful double helix over here. Bear with me. And we're going to get into translation. I really just want to use a different color. Hold on. We don't need all this. Okay, so in translation, we already know because it's written on here and we just reviewed it. Translation is the process of making protein from our RNA. Okay, so the idea of how this is going to work, you are going to have your mRNA. This box is in the way. I just wish that this box would go away. There it goes. So we have our mRNA. And it looks like this. Here's your code. A, U, G. What did we say? C, C, G, and A, whatever. What's going to happen with that mRNA is a structure is going to come and bind onto that first sequence. And it comes in 2 parts. It clamps down on it. So this big, bubbly, beautiful structure that I'm definitely giving it justice. I'm going to ask the name of this structure. Morgan. It's Morgan here today. Morgan. Morgan, what is the structure that binds onto it? We call it the machinery that's going to perform translation. All right, so this is going to come in and it's going to bind on. I'm just going to clean it up a little bit so that whatever I'm doing next will make sense. This is a major key component in our cells. We would consider it probably maybe like similar, akin to an organelle, but it's not membrane bound. It's actually made up of RNA. It's a ribosome. Okay, so this is a ribosome. Exactly. You're probably overthinking it. So ribosome, this is a ribosome. It's made up of ribosomal RNA. That's what it's made out of. Okay, so it's going to come and bind on to our start codon. Start codon is always AUG. So you notice I used a fancy word there. On our mRNA, this is known as a codon. I don't know why I wrote an R, but it's my name, codon. It's a 3 nucleotide sequence on our mRNA that codes for, well, what does it code for? An amino acid, okay? So an amino acid, right? So this will code for an amino acid, which are building blocks of our protein. So Adolfo, this is your question now. In the ribosome, so ribosome comes and binds on, and another structure is going to come bring in the amino acid. What structure brings in the amino acid? Beautiful. Yes, so that's going to be the tRNA. So the tRNA, and I'm going to draw him in a different color so we can see him. Okay, so the tRNA is going to have a sequence on his bottom. We'll draw him. He's this nice squiggly looking guy, whatever. and attached to his top, he has an amino acid. In this case, at the bottom of our tRNA, we have a opposite sequence. It's a complementary sequence. So if it's A, we would start with T, just as U, excuse me, now I'm already confusing myself. You guys know better than me, come on. You shouldn't have let me do that. All right, so at the bottom of this, we will have a U, and then what? Come on, guys. And then C, okay. So what is that called? That I just wrote out, that 3 nucleotide on the tRNA is not a codon, it's called something else. Anticodon, anticodon. So if that in the blue was a codon, this is the anticodon. The anticodon matches up with the codon. That's how it knows what amino acid to bring in. Okay, so this tRNA, and this is tRNA, brings in methionine. I don't need you to memorize every amino acid or what their structure is or anything like that. I don't need you to know that AUG codes for methionine, but it is probably the key, most important one of all of them, okay? So we always start with AUG and it always codes for methionine, always, okay? All right, so AUG on our mRNA matches up with the UAC on our tRNA anticodon, and that tells us to bring in methionine. I don't know what CCG codes for, but who cares? We do know that it'll match up with a GGC on our next tRNA, and that it's going to bring in some sort of other amino acid, right? In that next spot. Then these are going to create a bond. It is a covalent bond. What's the name of that bond? It is a covalent bond, but what kind of covalent bond? More specifically, you guys know this, peptide bond. This is where our amino acids link together. So if one, remember we draw all of our amino acids? Yeah, y'all thought you were done with chemistry? What a joke. Honestly, I can't believe you guys thought you were done with it. Okay, OH. Thought you learned it for nothing. Interesting. This is an amino acid, right? In order for that amino acid to link to another one, instead of that H being there, this will link like this to that next N terminus. This bond right here is our peptide bond. That's what's actually happening whenever we link methionine to another amino acid. Okay? Is this helping a little bit with seeing like the big picture of how this is all working? I hope it is, because I know it's a lot. And I know we're like still in chapter 6, but I feel like we can get this foundation down and it makes more sense to you because people do the worst on chapter 6 and 10 by far. And guess which chapters I have the most questions on the test from? 6 and 10. So we want to get that foundation on that. Unit 1, I spread them out evenly with the chapters, but it's not going to be that way from now on. Now I'm going to ask more about the stuff that is more important because it's foundational. That's chapter 6 and 10. Okay. All right. So you've been warned. Anyways, so we're getting an idea of what's going on with our peptide bond. This is all there again. There's a video of it. I wonder if I can. I just learned something. Hold on. I wonder if I can erase. No, it just draws. Okay. Whatever. Okay. So let's move on. We got from transcription, we got into translation a little bit, and let's see if we can answer the rest of our questions and then move on to our activity really quick. Okay. Good. Beautiful. A codon is 3 bases on. We didn't talk about it. It is up there in the light blue, but. So those 3 nucleotides, those 3 bases on our mRNA was originally coded by our DNA, right? So why aren't they called the same thing? In DNA, we call it a triplet. Well, once it's on your mRNA, it's a codon because that's what we're actually going to be reading to bring in our amino acids. So our codon is our 3 nucleotide sequence on our mRNA. 3 nucleotides or 3 bases. The bases determine whether it's A, U, C, or G. All righty. In what kind of operons are repressor proteins bound to operators unless a molecule that changes the repressor shape is present. I know this is a wild one. This is why we got past calling on people before we got to operons. Yeah, there's a reason. Yeah, give it your best shot, exactly. Now clean up a spot over here for some operons, because we gonna need it. Okay, it's okay. It's okay. Take a deep breath, everybody. All right. It's okay. Operons are easily, like I say easily the hardest thing in chapter 6. All right, so we're gonna go over operons. No worries. So in an operon, we have a DNA sequence. Okay. We have a DNA sequence, and it doesn't matter what the sequence is, but it'll be made up of 3 major parts. This is in your DNA. You will have a promoter region, an operator region, and then your genes that are associated with being controlled by that promoter and that operator. So there's, I don't know, 3 genes, gene 1, gene 2, and gene 3. There's 3 separate genes in the sequence. So this is an operator, or an operon, excuse me. It's in the DNA. All right, so you guys remember how when we're transcribing, we're going to open up the DNA and make an RNA single-stranded copy. All right, and the enzyme that does that is called RNA polymerase, right? So RNA polymerase always binds, whether you're an operator or anywhere else, RNA polymerase always binds to a region on that DNA called the promoter region. So the green here is our RNA polymerase binds at promoter, which in this case is our P here. Then we will open up the DNA and make our RNA. That's transcription. So we got to remember transcription in order to understand what's happening here. Okay. So it will usually bind onto the promoter, open up the DNA, and read all this stuff and make our gene product. This is our book, and we're going to make our mRNA from our genes. We don't make an mRNA from the operator. I'm going to tell you what the operator does. The operator is where a repressor binds. This is how all operons work. If the repressor is bound there, RNA polymerase can't get through. It's just like if you were trying to go open up your cookbook to get your recipe out, but there's a lock on it. Just whether there's a lock on it and you can't open it, or there's not a lock on it and you can open it and make a copy of the recipe. Just think of it as a lock. It's a physical barrier that makes it so that RNA polymerase can't read the genes and make RNA. Okay, so that's all operons. All operons work this way. Now, if you are an inducible operon, if you need to induce labor in a woman, is she already in labor? No. So if you need to induce, you started off, which means we can turn you on. We can induce you. Okay. So this, how we drew this picture, if you started off, what we mean is you're off because we're locked. The repressor is bound. The lock is on your cookbook. Okay. That's the inducible operon. The repressible operon you can be repressed because you started on. And I'll even put turn on, just to make this clear in case you aren't sure what repress means. Okay. So a repressible operon didn't have the red there. Repressible operon starts off like this. Oh no. Starts off like this. Repressible operon starts like this. And then after it's been going, now we can bind a repressor and block it. Now we can lock it. So it starts with the cookbook open, but then your body, your cell, notices you are making an awful lot of spaghetti and meatballs. We're gonna lock this cookbook. Okay, so that's what's happening. So think of it this way for repressible operons. Your body is basically making spaghetti and meatballs. And as long as you're eating spaghetti and meatballs, there's no buildup of spaghetti and meatballs. You just keep eating it and it's gone and there's no buildup. Once you start making spaghetti and meatballs and nobody is eating it, you've got a lot of spaghetti and meatballs sitting around, your cell can say, hey, there's spaghetti and meatballs sitting around. We're going to stop making this. Just like a factory. If a factory is making dresses and summer dresses, and it's summertime, people are buying summer dresses, well, they'll keep making the dresses so people can keep buying them. But if it's wintertime and nobody's buying summer dresses, why are they going to keep making summer dresses? And so they've closed down the factory until it's summertime again. Yeah, spaghetti warehouse, yeah. So same kind of concept there, right? And it's the same way with this. Inducible operon. The example that they gave of an inducible operon in our textbook, and one that you do need to know, it is in our learning objectives, is the lac operon. All right, so it was off, just like it is drawn up here. Lactose comes in, okay, which is a sugar. We need to break down lactose. It's not something we see every day. We only need to break it down when it's there. So when lactose shows up, it binds onto the repressor. Once it binds onto the repressor, it causes the repressor to change shape, like we often have with proteins. All right, chemical reaction, it changes shape and it comes off. And now we can unlock the cookbook. Think of the lactose as being the key to unlock the cookbook. Okay, it causes the lock to change shape. Keys cause locks to change shape. That's what happens there. All right, so the lactose is the key that unlocks it. In the case of the arginine operon, what happens is there is a guy watching around. He's not bound on. We're making arginine, which is the other one, the repressible. Arginine is an amino acid. Okay, as long as we're using arginine, it's fine. It's not building up in the body, just like our spaghetti and meatballs. Once we have too much arginine, or once we have too much spaghetti and meatballs, this happens. And again, in this case, we're taking the lock and we're using the key to lock it on. So what happens is this, after it binds with arginine, binds onto here and now locks on and blocks. Okay? So that's it.
So is it open or closed right now?
It's closed now because we were able to bind it on then. So it's just whether or not it was open. If you're open, you're making the product. If it's closed, you're not making the product. So if you are open, you're on. And if you're closed, you're off. Okay? So we can either start open and then turn it off, like we do with the arginine one, or we can start closed and turn it on. Open it.
How do you know if you're open or closed? I'm sorry.
Okay, so it's whether or not you're making the genes or not. And that depends on what type of operon you are. That's what you guys have to know, and you will get tested on this on the test. Okay, I might even tell you it's the lac operon, but I generally on the test will also say things like, it's the lac operon, which is an inducible operon. And if you know what an inducible operon is, then you will know if it's inducible, is a woman in labor, like, you know, if she has to be induced, she's not in labor, right? So if it's inducible, you can induce it. It was off, and now you induce it with lactose. Okay, are we tracking a little bit? So yes, so keep that stuff in your mind. Inducible and repressible is way more important to me than memorizing lac and arginine. Okay, yes.
When the repressible operon locks something, what is it locking again?
The transcription. So remember that the process that we have up here, yes. So whenever we have a repressor bound on here, I wish I could just erase the yellow, but it's not letting me. Whenever we have a repressor bound on here, the green can't move through. And what's the green? RNA polymerase. And what's RNA polymerase do? Transcription.
Okay.
Yeah, yes. So this is all happening on the DNA. The lock is being put on the DNA, the cookbook. That's why we use that analogy. Okay, are we good? Any other questions? Because again, this is one I know is tough, so I want to be sure we work through it.
Can I
Yes.
On the other side, the inducible, when it goes, it starts transcription.
So they all work this way. They all work this exact same way, whether you're inducible or you're repressible. If you're inducible, you start with the red on there. If you're repressible, you start with it off. And then eventually the red. So if you're inducible, you started with the red and we'll remove the red to turn it on. If you're repressible, it's open and you repress it by adding the repressor and you turn it off. But they both work with that same principle. It's just whether you started on or you started off. If you started off, the repressor was bound. If you started on, it's like this. Okay? Any other questions?
So the lactose turns it on
By removing the repressor.
and then the other one.
Arginine turns it off by causing the repressor to bind.
Are there other things that can do that? Do we need
Oh,
to know?
yes, but no, you don't need to know them. Just,
Okay.
I would focus more on inducible and repressible. We'll actually talk about an operon later on in lab. You won't realize it's an operon, but it is. We'll talk about one later when we get into transformation.
So the inducible will start transcription.
Yes, so by removing the repressor. So we call it an inducer, right? So lactose is an inducer. So because we had a repressor on here in the inducible operon for lac operon, okay, which is the most commonly studied operon in all of science ever. So we start off with a repressor bound, which is red for repressor here. And lactose comes in and is our inducer. When the inducer binds to the repressor, it causes the lock to unlock. So this is the first step, binding to it. The second step is removing it. And now because lactose bound to it, now it's gone. And now the green RNA polymerase can do its job. Okay? Any other questions? All right, I know it's a lot, but you guys are doing great, I promise. Okay, so that's that. Last question for chapter 6, missense mutations. Definitely know your different types of mutations. And we're going to very quickly go through our activity for chapter 6, and we're just going to jump through and do the Kahoot really quickly for chapter 8. Yeah, because we're running out of time, exactly. All right, so missense mutations. Definitely go through this. These are flashcard types of things, so it shouldn't be too bad. But the different types of mutations are missense. Missense is the one where the amino acid is changed. So if you had AUG, which coded for methionine, if you change to AUU, and now it codes for leucine, that's a missense mutation. If you went from AUG and it went to AUC and it's still coded for methionine, then that is a silent mutation. If you went from AUG, which codes for methionine, to UUG, which would be a stop codon, that would be a nonsense mutation. Okay, so those are the major types. We also have frame shift mutations, where we delete a nucleotide. So instead of AUG, CCC, GGA, now it's A, remove the U, G, C, you know, and it shifts everything over. It's like deleting a letter in a book, but shifting everything over to keep the word size the same. You couldn't even read the book after that. You open it to the middle of the book and it just doesn't even, that's a frame shift mutation. All right. It's always 1 or 2 nucleotide changes. A back mutation is if it used to be AUG, at some point it changed to AUC, but now we're back to AUG. That's a back mutation, changing back to the original. Okay, so this is the general types of mutations. So we'll be done with this chapter now. Here's our podium. It's a lot. That's why we spent the most time on it. Because I feel like chapter 8 doesn't make a lot of sense unless you really understand chapter 6. All right, beautiful. So it's going to pull up the thing to hurry up. It really takes its time, dropping its confetti. Congratulations. I don't want to poo-poo on your congratulations, but it takes its time. Chapter 8. I'm just pulling up chapter 8 so that it's here and then we're gonna go over our worksheet for chapter 6 really quick. Chapter 6's worksheet, I'm gonna make it bigger, hold on. So you can read it. All right, so this is talking about HIV. HIV is human immunodeficiency virus. It is a retrovirus. HIV, it talks about this up in this, and I only give you this sheet, but like it's as much as you would need to answer these questions, I promise. All right, so when we get down to the bottom down here, let me pull up my pen. I want pink. All right, so previous study discusses HIV. Its virus particles have a single strand positive sense RNA genome. Our genome and the genome of all cells are DNA. But this virus, viruses can have genomes made of RNA. Okay. So what does this mean in terms of transcription or translation when the virus enters the cell? A positive sense RNA virus can be read directly by the ribosomes and the tRNA. In that process of translation. So literally, when HIV gets it into your cell and has its little genome free in your cell, your ribosomes can start translating it. That's what a positive sense RNA virus does. It's a single strand, positive sense RNA. What that means is basically, effectively, for all intents and purposes, it is mRNA, basically. Okay, we're tracking? A negative sense genome would have to be turned, a negative sense, if your genome was AAA and you're a negative sense, you'd have to turn it into positive first. And then the ribosome can translate it. So that's what negative sense. So I know we introduced that in chapter 7, but now we have better context for what that really means, okay? So is this positive sense, the ribosome can read it. If it was negative sense, we'd have to turn it into positive sense, and then the ribosome can read it, okay? All right, so that's its genome. That's what the virus actually has in its little happy little virus particle. It is an envelope virus, and here it has a single-stranded RNA. It's positive sense inside of it, and it has an envelope around it and has spikes on its outside to get in. All right. Study mentions that HIV produces reverse transcriptase. What does this allow the virus to accomplish? Anybody know what that means? Yes, so it does. It does copy. If transcription, right? Remember how we talked about transcription? If we replicate, we're going from DNA to DNA, right? Making a DNA copy. If we transcribe, we're going from DNA to RNA. So what's reverse transcription? RNA to DNA. So we're going to take that single-stranded double RNA genome and reverse transcribe it with using that enzyme, reverse transcriptase. Remember that ACE is an enzyme. And usually the ACE means it does what the word is before. Collagenase is an enzyme that breaks down collagen, for example, okay? So DNA ligase is an enzyme that ligates or joins together DNA, okay? DNA polymerase is an ACE enzyme that polymerizes or makes DNA, right? We're tracking with that. So reverse transcriptase, reverse transcribes RNA. This is an enzyme that comes with HIV. It is made by HIV, and it turns RNA into DNA. Our cells do not have reverse transcriptase. It's actually key for us understanding how the COVID vaccine works and why I am so sick and tired of undereducated people telling me how the COVID vaccine changes your DNA. How? How does mRNA change your DNA? Anybody? It doesn't. Exactly. Because you cannot reverse transcribe. Period. Okay? You cannot. Even people with HIV can't just take any mRNA and turn it into DNA. It's very specific. Okay? So even if you have HIV, you ain't turning the COVID vaccine into DNA. And even if it did turn it into DNA, we'll first turn it into DNA, and then another enzyme called integrase, also that comes with HIV. So it's HIV reverse transcriptase and HIV integrase, both of them made by HIV. Integrase, also ACE enzyme, that integrates the DNA into the host genome. This doesn't just happen naturally. We have to have enzymes that drive this. And they come with HIV. You don't have it in your cell. So you not only need reverse transcriptase to turn it into DNA, but you need integrase to insert it into the DNA. Okay. All right, we're tracking with that. All right, so this is happening with HIV. HIV infects certain cells. So only certain cells in your body even have reverse transcriptase and integrase in them when you have HIV. And specific cells, they're your CD4 positive T lymphocytes. You probably heard that and maybe known that about HIV, but you don't know what your CD4 positive T lymphocytes are, but you will. That'll be unit 3. And you'll learn a little bit about it in physiology if you haven't taken physiology, but we'll get into it more in depth on how it affects your infections and things like that. So they are immune cells. But that makes sense too. So HIV is infecting immune cells, which is why when HIV infects immune cells and kills immune cells, why you get immunodeficiency, which is AIDS, acquired immunodeficiency syndrome. And as it turns out, your CD4 positive T cells, the cells that HIV infects, are the captains that literally are the captains that tell all of your immune response what to do in any immune response. So it's why we get widespread opportunistic infections associated with HIV. Okay. All right. So reverse transcriptase. What role would replication play in the HIV viral life cycle? Oh my gosh, this is a mess. In this case, let me remove all this. So in this case, replication. So once we get the DNA incorporated in, so we started as RNA. Really? This thing works different than my old one. We started as RNA. We became DNA. We inserted it into the host genome. So now we are the host genome, the normal genes, and we inserted the little HIV DNA version in. Do you guys remember what the term is for it once that's inserted? You should, since all of you missed it on the test, except maybe 1 or 2 of you. This is provirus. Yeah. Transformation is only if we turn it into cancer as a result of this. Okay. So this is the provirus. It's not the virus anymore, right? It's not got the envelope and the capsid and all that. It's just the genome. It's the DNA version of the genome inserted into the host genome. Now it's the provirus. Remember how we call prokaryote before nucleus? It's the same kind of idea here, before the actual virus inserted into the host genome. So now when the host replicates its cell, you're also making a copy of that built in with your genome. So that's where replication plays a role in viral life cycle. Okay. All right, study mentions reverse transcriptase is the best target for our treatment. Why would that be an excellent target? Because obviously our cells don't have reverse transcriptase. It doesn't affect your cells negatively. In fact, most anti-HIV medications, including PrEP and STEP, which prevent HIV infection, are going to be targeting reverse transcriptase or integrase, those 2 enzymes, which our cells don't have. And so it shouldn't have toxicity to our cells if we block that. In fact, when you do studies, we read studies about medications that affect reverse transcriptase and integrase, you'll find that it's not going to be really harmful for you to take those medications. And they are quite effective at preventing the virus from replicating. We use it for treating HIV. HIV can't be cured, right? So once it's integrated into your genome, that's forevers. That's for keepsies, okay? It's forever. HIV is forever. There's no curing HIV, but there is treating it. So we can give you the antivirals that basically stop reverse transcriptase and integrase. And now the cells that you have in your body have that provirus in them forever, but it can't make new ones to infect new cells. So we stop the spread of that virus that way. Okay. And that's how you can get people's viral loads down to 0. If your viral load is 0 or undetectable, does that mean that you are HIV negative? No. If reverse transcriptase can convert RNA to DNA, how might this be used in research and medical advancements? So imagine that you have a virus that can insert genes into your genome. Now imagine that it's inserting HIV genes into your genome. That's a problem. But now imagine you replace those HIV genes with a working gene when you previously had a defect in your genetics. Imagine you can't make your own enzyme that's important for making clotting factors and things like that. But now I can insert that gene into your genome using HIV. And that is how gene therapy works. We're going to talk about a paper next briefly again, because we're going to play a couple questions on Kahoot, and then we're going to jump right into the paper review for chapter 8 very briefly. But it talks about gene therapy for sickle cell disease, right? With sickle cell disease, you have a genetic defect that causes misshapen red blood cells. That leads to basically damage of the red blood cells, splenomegaly. The spleen has a hard time filtering through. It has a problem with carrying oxygen. It can be deadly to have sickle cell disease. If you have the sickle cell trait, which is just having one version of the gene, you can still have disease associated with that. If you have two versions of the genes, it can be deadly. But we can correct that by infecting somebody with HIV, but replacing the HIV genes with the correct gene for your red blood cells, basically. And it works. And it costs $1.5 million per person. And your insurance will not cover it. That's what that other paper is about. So we can start to see how many of you guys got that sink in your life. We can cure diseases. We can cure. You have Huntington's disease. You guys know what Huntington's disease is? It's like a genetic mutation that is, you'll die by the time you're 27. It's only in women. Okay. We can cure it.
For the right price.
For the right price. Yes.
But you have to infect them with HIV.
It's not HIV anymore. It's not HIV anymore.
Oh.
It doesn't have the HIV genes. It only has what it needs. Yes.
It's like you gotta pick which one.
Mhm. Yeah, so no, it is not HIV anymore. Remove any of its gene. It can't make spikes anymore. It can't make its capsid anymore. It is only making reverse transcriptase and integrase. And then it has that good gene. So it can't make the virus anymore. It is only doing that. We're using it as a shuttle. It's like hollowing out and putting in good stuff instead. So it's not going to be the virus anymore. It can't be the virus anymore. It can't even revert back to being the virus. It doesn't have the tools that it needs. Okay. So that's the idea. And that is how gene therapy works, and it does work. And it is lifelong. It cannot be sent down to kids later on down the line life, because we're only treating basically your bone marrow. So in the case of sickle cell disease, cells that are affected are your red blood cells. Where are your red blood cells made in your body? Your bone marrow, in your bone marrow. So we can literally do a bone marrow transplant. We don't even need a transplant. We can take your bone marrow out of your bones, infect it with our engineered virus that isn't HIV anymore. It's a better version that has the correct gene, and then mix it in with your bone marrow and then reinfuse you back with your own bone marrow. And now you've got the gene that works. And your red blood cells that you make are new. They have that gene in them. Amazing technology, and it works. Problem is, when you start inserting genes into other people's genes, you can also have side effects that we didn't necessarily predict, and that we couldn't predict in animal models. That does seem to be relating to side effects from the gene therapy. We also don't know long-term effects of changing somebody's genetic sequence. So then we have negative effects associated with that. So that is what the study was going to be talking about for chapter 8. If you want to read through it and learn more about it, I highly recommend that you do, but we're probably just going to jump into chapter 8's Kahoot. But this is treating basically sickle cell disease and talking about possibly treating other diseases with gene therapy here. This is somatic gene therapy. Germline is if we can affect the gametes. That's the only way you can pass it down. Because if we're just affecting your bone marrow, how does that get transmitted to your kids? It doesn't. All right. And so in order for it to get transmitted to your kids, we have to affect the gametes. The only way we can affect the gametes is when the gametes form in your body. When your gametes form in your body, when you're a fetus. And so we would have to affect a fetus. And so I'm sure you can understand what the ramifications and issues might be associated with that, right? Potential benefits of this therapy and why that might be significant in this context. Obviously, this can save lives. Here, we're talking about sickle cell disease that can save lives, but we could eventually be using this technology for a lot of other mutations that could be deadly and things like that. The estimated cost is $1.5 million. So when we're talking about this is a major risk. If you guys don't know about sickle cell disease, it's primarily found in African population, people of African descent. And so, and it's very painful. Yes, it causes very extreme bone pain because of the process that's involved in making the red blood cells, again, splenomegaly, and it can become life-threatening. So,
People who treat the pain.
yes, and so if we're talking about people that live in third world countries, how are they gonna pay for the $1.5 million to be cured of this disease? And who's gonna pay for that?
Lucky in the United States, though, too.
People in the US have it too, but they're gonna be probably, so again, people in the US have it. You know, what percent of our population, of our low income population is Black? Are they gonna be able to afford this treatment when they're on, you know, government healthcare plans like Medicare and stuff like that? No, so this leads into that bigger question of, you know, insurance and, and insurance companies and what's funded and what isn't. Now, like I said, it's not a perfect painted picture. There are issues associated with this. It comes back also to that same concept of people read in also. You might hear like, oh, they're infecting, you know, me with HIV. It'll cure my sickle cell disease, but they're infecting me with HIV. I'm not going to do that. And that's what they hear. It's the same rhetoric that we're getting with vaccination right now. It is uneducated and it is making decisions for people that could be life-saving. And instead now people will die from COVID and measles and things like that because they're not getting vaccinated when they should. So my mom, I talked to my mom this weekend. My mom is not a science person. I feel like she's one of those people that's lucky to have had any education that she did have. But she has an associate's degree. I'm not poo-pooing on an associate's degree because many of you guys are getting an associate's degree. It's definitely good. But she got an associate's degree in computer science back in early 2000s, which is almost irrelevant now, right? And so, and she also hasn't worked in a very long time. So that's what her education background is, just to make it clear, right? And she calls me up and we were having discussions and one thing leads to another. And she's like, oh, well, you know, first she starts off with COVID can change our DNA. And so I gave her the same talk that I give you guys, okay? And then she's like, oh, well, okay, well, you're arguing against that, but it causes heart disease. The COVID vaccine does. And I was like, okay, so now we're getting somewhere. So let's have that discussion, shall we? Okay. Is the COVID vaccine linked with causing heart damage? Does anybody know? Okay, so it has been shown that we have an increase in coronary artery disease. I want to be specific heart damage, coronary artery disease, okay, which leads to heart attacks. But the COVID vaccine may be linked to that. There is no evidence proving it or showing actual cause, but there is correlation. All right, there is. It seems to be small, but there is a correlation. And so you might be sitting here thinking, hearing me, and you're like, the COVID vaccine can cause heart attacks? Maybe, yeah. Do you want to talk about what COVID causes? Because that's where people are forgetting. We are not talking about the COVID vaccine versus not having the COVID vaccine. We're talking about the COVID vaccine versus having full-blown COVID, unvaccinated COVID. How many people in here, you had COVID or somebody immediate in your family had COVID? Raise your hand. Look around, folks. The risk for death from heart disease, from having full-blown unvaccinated COVID is much higher than it is for vaccinated people, period. That's what people are forgetting.
What about if you got it before the vaccine was developed?
If you got COVID before the vaccine was developed, you're at higher risk for heart disease.
I was gonna say, especially because I work at the hospital, I feel like a lot of people forget the COVID thing. I literally had a patient last night on ISO for COVID. I was just like,
Yeah, people forget how serious it was. Do you not remember how scared we were? Like, do you not remember how many people were dying from COVID? That wasn't a joke. We had healthy people dying. So if you want to go back to that, I mean, you can't. You're vaccinated. This is the rhetoric of people who are vaccinated protected, poo-pooing on vaccinations that they have. It's wild.
What's the story on if you had the disease and then you get over it? Do you produce antibodies for it, or is that
Not protective. So we don't think it's protective. There's no evidence that it is protective. It might be at the level of the vaccination, but not like completely protective. So obviously you can get COVID over and over again. We think that having had it originally may be associated with some protection equivalent to having gotten the vaccine. Remember, vaccines are designed to help your body create memory and things like that. Having a disease doesn't do that. So a lot of people will be like, well, the chickenpox does. Yes, chickenpox does do that. Measles, if you get the measles, you can get the measles over and over and over and over and over again. Every time you get the measles, you have a 1 in 1,000 chance of dying. And having had it previously does not protect you.
Chicken pox different.
It's just how your body reacts to it. Mhm.
So-
Different virus.
Your body reacts to every antigen completely differently than any other antigen. In fact, it's so different to your body that we think of like flu vaccines as being a thing, but every flu vaccine that you get, your body sees it as completely different as COVID is to the plague. Your body doesn't know that they're related at all. It sees them as different. So your body reacts differently to every flu virus. And that's why we get different strains. Might as well be a different virus to your body. And so there's that. So how your body reacts to things, it just depends on how it sees it and what signals it's getting and all that sort of stuff. And the measles virus has stuff in play that prevents your body from creating memory. In fact, we talked about in one of my other classes, having the measles. Did we talk about this too? Having the measles. Having the measles predisposes you to having MS. I think we did talk about this. Yes. Yes. You have the genes for MS, but having the measles activates the genes. And now you actually have MS. And so we have links like that as well. So it's just different. Every virus is different how your body reacts to it. So the next time that you have that discussion with somebody about any vaccine ever, please just keep in mind, we are not talking about the vaccine versus being healthy. We're talking about the vaccine versus having the disease. So now you can get COVID and you get a cold or flu-like illness because we've trained our body to react appropriately to it. It's not perfect. We'd rather have a vaccine that prevented you from having the sickness at all, but it is what it is, right? We can talk more about that because I had this whole discussion with my mom, and I know that it's something that a lot of people hear, and you're watching it on social media, and it's getting around, and all these people are telling you this stuff. Read the articles yourselves, you guys, because I promise you the people that are saying on social media didn't read the articles. You can, and you can do better. You
So
can know better.
monoclonal that you got when you had COVID, it's just coincidental, you got sicker.
Yes. So, and that just depends. Yeah. And that depends on how your body reacts to the monoclonal antibody and whether or not it's actually giving protection. I will say this, there is a lot of evidence to soothe some of you guys freaking out. Like I either have heart disease or I have COVID and die from it. Like you're already vaccinated, right? I think everybody in here probably is vaccinated. It's almost required now just to be in society. For a while, I think it was required that most people were required to go back to work, you had to have the vaccine. That's how it was when I was working. But everybody in here has been vaccinated for COVID, right? If you've only got the 2 shots that were required, get the 3rd one. And this is why you just told me that it can cause heart disease. Having the 3rd one decreases your risk. So if you've only gotten the 2 and you're like, I don't know if I want to get the 3rd one because heart disease, get the 3rd one because the 3rd one reduces that risk. Trains your body better. I don't know enough about the molecular aspect of it, but it said it basically is another exposure to that antigen, letting your body know this is an antigen that we need to respond to this way, as opposed to freaking out from it and potentially leading to inflammation in the heart. That's the best explanation I've got for it. All right. It's so complicated that like even me having had training in that stuff, it's just too much for an individual antigen for me to know about it. Okay, we're tracking. Get the 3rd booster if you haven't, and you will reduce your risk for heart disease that you're so scared of. Tell your parents that too. All right, so that's it. We're not going to go over the 3rd Kahoot. Again, if you ever want to go over the 3rd Kahoots, we're not going through it today. I already got to everybody that was here, yes? Everybody got their points if you got called on today. We're not going through the 3rd Kahoot. If you want to play it, look for it in your modules. It's lecture activity, chapter 8, and you can play through it for studying, okay? Otherwise, we're going to head up to the lab, take a break. I'll see you guys at 11 in the lab. We'll go over PCR later. Because we definitely need to talk about PCR. I feel like that's one thing that we need to talk about. We'll do that later.